Welcome to Almost Here, Round the Corner Future Technology podcasts with Richard Jacobs. Future technologies poised to transform our lives for better or worse are the focus of this podcast. Almost here means these technologies are now here and starting to be used, or just around the corner, from Bitcoin to artificial intelligence, 3D printing, blockchain, virtual reality, and more.
Hi, this is Richard Jacobs with Future Tech Podcast. I have Dina Zielinski from the New York Genome Project, or NY Genome. Dina, how you doing?
I'm good. How are you?
Good. Thanks for connecting. I appreciate it. Can you tell folks what you're working on over at Columbia?
Sure. So I work in an academic research lab in Yaniv Ehrlich's lab, and we're at the New York Genome Center and are affiliated with the Computer Science Department at Columbia University. And we work on a bunch of different projects. There's never a dull day. I've been in the lab for 6 years, and I can never come into lab and say there's nothing to do. We've always got a very diverse number of projects going on, from human genetics to storing data in DNA, which is one of our most recent publications.
You said storing data using DNA?
Yes. So we recently published a paper where we were able to store a bunch of files in strands of DNA, and then we were able to sequence the DNA and fully recover the files.
Wow. Well, I know the human body essentially does this. Can you talk about how the human body does this? And it sounds like you guys took inspiration from that to store data. Can you go over the two and contrast them?
Exactly. Well, more than the human body, it really just comes down to the DNA molecule itself. It has had a lot of time to become kind of an optimized system. It's very stable. It's very dense. You've heard stories probably of recovering DNA from cavemen from 100,000 years ago. And granted, that DNA is quite fragmented, but we're still able to sequence it and recover some information about that individual. And so we decided, among some other collaborators too, to store data in DNA. And this is not entirely new. People have been working on this for the last few years, including George Church at Harvard. And so what we wanted to do is sort of improve on the current methods. And so we took one approach that uses a different algorithm called fountain codes. And by using this algorithm, we were able to store more information than previous studies and also recover the data without any errors.
Wow. How much data can be stored in a single DNA molecule?
Well, the theoretical maximum is about 2 bits per single DNA base. So you have your A's, T's, C's, and G's. But this is not actually realistic because not all DNA molecules are created equal. There are some DNA molecules that are difficult to synthesize and sequence. And this generally consists of DNA sequences that are highly repetitive. We call these homopolymers. So if you have a bunch of A's in a row, the synthesizer and the sequencer do not like these. We also have some issues with GC-rich regions. So these are regions of DNA that contain a lot of G's paired with C's. And that's because there are 3 hydrogen bonds between those. So it has, it makes it difficult to both sequence and synthesize. So we try and avoid these GC runs and these homopolymer runs, but that also limits the amount of data we can store in each individual oligo. We basically will chop up a bunch of files. When you compress data on your computer into, say, a zip archive, you're basically just kind of converting that into zeros and ones. And so it's the way, our approach basically is just a different way of packing up those zeros and ones and balancing how much data we can store while also adding some redundancy. That's really what data compression comes down to, is you have to add some redundancy to your data, but you also want to limit how much redundancy you add because that will take up space, storage space. And so once we take those zeros and ones and package them up into what we call droplets, that's where the DNA fountain gets its name, we can then simply convert those zeros and ones into A's, T's, C's, and G's that's actually the easiest step. You basically just go from A to 0, 0, T to 0, 1, G and C to 1, 0, and 1, 1, respectively. And so once we have that, we can send a list of sequences that are 100 nucleotides long, so 200 A's, T's, C's, and G's, and we can just send that to a company that synthesizes DNA, and then they'll return this DNA in a tube, and then we simply sequence it and get all our data back.
Why bother doing this? We have computers, we have billions of transistors on a chip. Why bother using DNA to store information?
So as you brought up, and when we talked a bit about how long DNA can last, it can last orders of magnitude longer than your standard hard drive or your server. DNA would be stable, cold, and dry for hundreds, even thousands of years, whereas you have to replace solid hard drives every few years, maybe 10 years. And this gets really expensive. And the more you read and write these hard drives, the more chance of your data being corrupt. So DNA is a really elegant solution to storing data long term.
It's amazing. What about the density of the storage? Is DNA able to store data more densely than current transistors and chips, or less densely?
Yeah, absolutely. So to give you sort of a toy example, like an extreme example, most of the world's data has actually been generated in the last few years. This is a big function of social media and, I guess, oversharing data on the internet. But a 1 terabyte hard drive right now currently weighs about 150 grams. And we were able in our study to fit 215,000 times as much data in 1 gram of DNA. So basically, if you store data in DNA, you could fit all that data in the back of a pickup truck.
Wow. Really?
Yeah.
That's amazing.
Absolutely. Yeah, so it's a little more efficient.
That's crazy. So DNA is thousands of times more efficient. Well, almost 1 million times. Yeah, I guess 1 million times more efficient.
Yeah, I mean,
how it stores data?
215,000 times more data in just 1 gram.
Wow.
So I would say so.
It lasts orders of magnitude longer than solid-state drives, etc.
Yeah, yeah. I mean, it's very robust, and it'll be stable for many years in just a refrigerator. Even at room temperature, it would be stable for quite a while.
How can this be? I mean, what is it about DNA that allows it to do this and have these amazing properties?
It is just a very simple, stable molecule, and it's mostly we've learned just by being able to sequence DNA from those cavemen hundreds, thousands years ago, and just by storing DNA in the lab, we've been able to show that it is just highly stable as long as it's kept relatively happy, dry, and cold.
Can you store DNA using the code that is already there? Meaning, can you store information using a living organism's DNA as the coding or encoding? Or do you have to make your own custom sequence to store data that you want?
So right now we have to use synthetic DNA. And this is actually the most expensive part and kind of the bottleneck to really making this useful, to making DNA storage a thing. To give you an example, in our study, it cost about $7,000 to write the data, to write the DNA, and about $2,000 to read it, to sequence it. So it's still pretty costly. But that's because when biologists request synthetic DNA, you want it to be of very high quality. You want to know that if you ask for an A or a T at a specific position, that molecule is going to actually have an A or a T at that position. So it just takes up a lot of machine time to generate these high-quality oligos, they're called. And that's why we stick to about 200 bases, because if you get longer than that, the quality goes down with both reading and writing the DNA. So
when you're making these custom oligos or DNA, are you going to be able to make certain sequences that repeat? And what will make it cheaper and more scalable in your mind over the next, let's say 5 to 10 years, so that it can become widespread?
It really just comes down to using basically a quick and dirty synthesis. right now it's a very quality-driven process, and if we can, make sure that our method can handle this quick and dirty synthesis, then it would definitely become a reality. And we sort of demonstrate this in our study in that we made many copies of the DNA that was sent to us from the company. And we do this using PCR, so we can make, many, many copies of that original amount. Because one issue with storing data in DNA is that DNA is finite. What you get in the tube is what you get. But we can use the polymerase chain reaction to amplify this library. But by doing PCR, you're introducing errors. That's just part of the DNA copying process, part of the one of the flaws of the enzymes, the polymerases that are used. And so we were able to show that even by introducing errors, by making deep copies of our library, we were able to fully recover the data. So it will just come down to using quicker synthesis that won't generate as high quality sequences. But as long as our decoder can handle it, then we shouldn't have a problem. So I think most of the work will need to happen there.
So what do you see? What's the trajectory in terms of cost and speed to do this over the next few years? When do you guess it'll become commercially viable?
So I think it's going to require some larger scale interest. And I know that there are some companies who are exploring DNA storage. When those giant computers came out to store data, they were the size of a room. And it took a lot of research, a lot of money to get down to USB drives we use today. And so I think once this sort of gains widespread interest and more research is done into sort of pushing how much data we can store and into synthesizing DNA more cheaply, I think it will become a real possibility in 10 years or so. Still be costly, but again, if we can reduce those synthesis costs, that would be huge. And DNA sequencing costs continue to drop. They've been dropping exponentially, so soon enough that will be basically a non-issue. And we envision long-term storage, so you would only need to sequence the DNA if you want to access it, but it would be very stable to just store it long-term.
If you knew an organism's DNA sequence, could you store information without changing their sequence? Could you use their sequence as a cipher and still store data on the organism, or use their own DNA as a code to decode data that's unique to them?
So are you asking if you could store data in an organism's genome, like a human genome, for example? Yeah, so
right.
This is actually one of the questions we've gotten. It's very interesting. My personal opinion is it's definitely not worth the risk. Your DNA already tells sort of your family history. And we had some questions about, can I store maybe some family photos in my DNA to be passed on to my relatives? And it's a very cool idea, but it's like, half of your genome is repetitive. It doesn't necessarily do a whole lot, so we think. But I don't think it's worth the risk of inserting some family photos in your DNA, wherein you could just store those family photos in synthetic DNA in some freezer. So I think it's a bit of a stretch. I think it could be done, just certainly not worth the risk.
Well, what's the bad joke in all that? It's the true selfie.
Absolutely. Taking selfie to a new extreme.
Yeah, interesting. What has your research revealed to you about either the human genome or animal genome or any genome you've studied now that you see its capacity to store data? I mean, how much data does it hint at that is already being stored by our genetic code maybe we didn't know about?
Yeah, so one thing I've learned is that we still have an incredible amount to learn. The human genome reference sequence was only completed about 14 years ago. And we've certainly learned a lot. We've learned a lot about different diseases and traits. But even those simple, what we call Mendelian disorders, many of them still remain unsolved. And I've worked on some rare genetic disorders, complex disorders like Parkinson's, and it's just endless, the amount of work it's going to take, the amount of genomes it's going to take to understand these different traits and diseases. But in terms of how much is going on there, I mean, you've got 3 billion base pairs. It's a lot of data, and that all of that DNA just fits inside a single cell. It's pretty incredible how much information is in our DNA. And, only a percent of it encodes proteins, but we're learning more and more about those non-coding regions, and we're learning that they actually do play an important role in different traits and diseases, which is why I would counsel against storing family photos in those regions.
Right. Are you able to give an estimate, a translation of how much data is within us? Is there any way to say it's as if we have 10 exabytes of data, or is it not? Has anyone tried to do that, or is it not possible?
Not to my knowledge. No, I'm not really sure. If someone has looked specifically into measuring the amount of data, but one thing that we do know is that it's incredibly dense. That there is, in fact, a lot of information encoded in those 3 billion base pairs.
Yeah, that is. It's amazing. Is there anything that you found that can store data even more densely or better than DNA, RNA, anything else in the body or in living organisms that's an even better potential storage avenue?
There has been some work into synthetic polymers, but as in terms of naturally occurring molecules like DNA or RNA, I don't think it gets better than DNA. And
what does that tell you?
Store an RNA is that it's not as stable as DNA.
Gotcha. Okay. Is there any particular organism whose DNA seems to lend itself to storage better than others, or is it just DNA is DNA and it works?
Yeah, it's really just about the DNA molecule itself, not really organism specific.
Well, I was going to ask you, any other surprises, or what surprises have you had working on this project? What have you learned that you're just shocked to learn or surprised to learn that we haven't talked about yet?
So I have to be honest, the day-to-day in research, it's kind of just like any other job. It's very cool. I love it. I love continually learning, but it's very rare that you have these sort of eureka moments. It's very cool when things work, but often when things work, we're kind of, we question it. We're skeptics. As a scientist, you have to be a bit of a skeptic. So I would rarely say that I'm ever shocked in the lab, just maybe pleasantly surprised when things work.
Okay. And then, feelings that you've gotten from working on this, have you been so amazed that it's made you question anything, or has it reaffirmed any beliefs that you had, or you just think it's a fascinating thing that you enjoy working on?
Yeah, I think the latter. I mean, working in science, you just get to make a small dent in our understanding of the world around us. I mean, science is sort of the foundation of our deepest knowledge, but it's always constantly changing. But it's really amazing to just even make a tiny contribution into understanding the way things work. And oftentimes things sort of affirm prior knowledge, but sometimes you're surprised and you sort of question everything a little bit. But generally speaking, yeah, I think mostly we are just pleasantly surprised by the things we've been learning, especially about our own genome. And just to go back to your question about, I think maybe I misunderstood your question about how much data the human genome would represent.
Right. I
mean, if you take those 3 billion base pairs, you'd probably get definitely a few hundred megabytes of data. Since, as I mentioned, every base theoretically can be encoded by 2 bits.
Okay.
So yeah, a lot of data in there.
Any commercial applications that people are looking for beyond storing data? Any particular applications that you're being asked about?
You mean in addition to that study?
Yeah.
So I guess most scientists, or I guess I don't know if I can speak for everyone, but with basic research, our goal is really just in trying to better understand things. It's rare that we try and commercialize things. We're all about sharing. Even to give you an idea, we recently, I think it was about 2 years ago, we developed an application to help scientists in the lab to aliquot to pipette samples. This can be very tedious because one of the rites of passage in molecular biology is learning that you're just combining minute volumes of clear liquid. So it can be kind of maddening when you're doing this for many samples. So we developed this web-based app called iPipette. And certainly we could have tried to commercialize it, but we made it completely open source and shared it with the community. That's just kind of the way we operate. And so
that's great.
Yeah, we're all about sharing.
What does it do? What does the app help you do? Yeah.
Yeah, so you could check out the website. It's ipipette.teamehrlich.org. And what it does is it will illuminate wells of standard 96 or 384 well plates in the lab. And so you can upload a simple Excel file saying, I need to take this sample from well A1 and pipette it into well G1 in a different plate. And so it keeps the biologists a little bit more sane. Basically, the goal really was to improve reproducibility of different experiments. And we also had some struggles with top-of-the-line liquid handling robot and found that it was much easier if we just took the reins and combined our samples ourselves.
What's the difficulty in pipetting? Is it just hard to hold your attention on what you're doing? And
yeah, I mean, you have to be very focused, but we're human, so even if you're very focused, you might pipette sample into the wrong well. And so if that well is illuminated, it definitely decreases the chances of someone making an error. And I've used it myself. My background is actually mostly in wet lab. It's only in the past few years that I've become more of a computational biologist. So I definitely understood the woes of tedious pipetting. It can be very stressful. You definitely have to focus. So we hope that this would help people keep more sane in the lab. And we've gotten some great feedback for it.
Hey, okay. All right. Any final words or anything you want to say about the DNA storage project? Or you think we've covered pretty much all the bases?
No, I think we've covered the important parts. I think it really is a possibility that this will become a reality. It may be that you won't know that your data is being stored on DNA. For example, if you use any of the Amazon Web Services, it could be that instead of your data being stored on in a server farm, it'll be stored in a freezer, sort of a freezer farm. That's sort of what we envision for DNA storage.
So the data centers of the future, instead of being just rooms full of computers, they could be a big refrigerator with thousands of vials of DNA.
Exactly. Yep.
Crazy. Sounds like the Matrix. But okay. All right, Dina. Well, thank you for coming. It's been very interesting, and I appreciate your time.
Great. Thank you.
You have been listening to Almost Here, Around the Corner Future Technology podcast with Richard Jacobs. Subscribe to this podcast, both to review, to discover more future technologies that are poised to transform our lives for better or worse, such as Bitcoin, artificial intelligence, 3D printing, blockchain, virtual reality, and more.